Hi, my name is Maximilian Lombardo, and this is my video interview for the Treeway Summer Challenge. I want to first start off by saying why I want to do the challenge. I think that what you guys are putting together is very cool, in that you're combining people with many different backgrounds in order to use big data to try to create physiologically based models for ALS, and hopefully for the people involved to create something that will lead you towards a cure. Uh, I think this is very cool because you're going with the trend of big data, using big data, which is kind of everywhere in our lives right now. And I think it's good also to get not just mathematics people involved and computational biology people involved, but also chemistry students and medicine students. And I think an interdisciplinary approach is the best way to tackle some of these big problems that have uh, been unsolved so far. So I think that my background would be very interesting for you guys for this challenge. Right now, well, in undergrad I studied biology at Siena College in New York. Uh, there I did biochemistry research on a protein called GRK2 for a couple of years while I was studying. Following undergrad, I started a master's program in biotechnology at Columbia University. While finishing up my master's program, I ended up joining a lab at the Columbia University Medical Center where we do translational drug development for non-Hodgkin's lymphoma. One of my projects specifically deals with a therapeutic which is thought to activate the unfolded protein response, which is basically a stress related pathway, and we're utilizing the activation of this unfolded protein response to selectively kill cancer cells, and specifically our models of non-Hodgkin's lymphoma. I think that this may be very relevant for ALS in particular, because a lot of neurodegenerative diseases are associated with protein aggregates or RNA aggregates. For example, Alzheimer's obviously, is associated with, I think, the tau mediated or tau protein, and those aggregates are perhaps its prions. But I think ALS is also associated with RNA toxicity, and this would be actually specifically from the C9orf72 repeat, expanded repeat, which is found in a large portion of familial ALS patients. So I think that my background uh, in protein stress response pathways and cell stress response pathways may be a big asset towards modeling a cellular response towards the stress that might be caused in an ALS cell from RNA mediated toxicity. Additionally, I think my background is pretty useful for the challenge because I have some experience in analyzing next generation sequencing data. So for a similar project that we've been working on in this lab, we have developed a cell line that is resistant to the therapeutic that I was talking about earlier. And this cell line, we did next generation sequencing on it, and in order to kind of sift through the data, I learned the R programming language, and I've been utilizing some techniques to analyze the data, because it's a huge list of genes. It's 25,000 genes, but we have to narrow it down to just a few genes to say what is causing the resistance. So there I've obviously been doing like differential expression analysis, but I've also used a few other packages, like one is called the Viper package, which kind of identifies a transcriptional master regulator, which is responsible for larger changes within the cells, and this is more of like a systems biology approach. In addition to that, I took a class this past semester which was aimed at using our programming language as a platform for teaching the statistical methodologies which are used in gene expression analysis, in addition to like some other like chip data analysis and other high throughput sequencing data analysis. So I think that my background, and I think that the most useful part that I can play is that I can kind of speak multiple languages, if you will. You know, I have obviously very good molecular biology backgrounds, and I'm involved in translational research, but I also understand what goes into analyzing data in a deeper sense. And I think that I can really serve as a bridge between those two different worlds. And I think I hope that you see my value as being in that as well. Um, yeah, and I think that's also what I am in general. You know, I get along with many different cultures very well. I'm a dual citizen of the EU and the US, so I always kind of consider myself a citizen of the world, maybe kind of a jack of all trades. Yeah, that's kind of um, what I am. In addition to that, I think, well, actually, I think that's kind of it. Sorry the video is not 10 minutes long, but feel free to ask me any more questions if you have them. Okay, thank you.